This is section 12.4, and it is about mutations. So our key concept for this section is what are mutations. So that's the first thing we'll talk about. A mutation is a change in the genetic material, and there are several different types of mutations. We're going to talk about gene mutations and chromosomal mutations. Okay, a gene mutation is when you have a change in one single gene. Okay, that's a gene mutation. And then a chromosomal mutation is just like it says, is we're going to have some sort of change in the chromosome. Okay, you'll remember that genes are located on chromosomes, and they involve series of nucleotides. So sometimes a gene mutation can involve like just one, or it can involve a few nucleotides. Okay, a point mutation is when you have a area on the DNA sequence where it's a single point is changed. So in other words, you're changing out a letter. And sometimes the letter may be substituted with another letter. A letter can be inserted in there, or a letter will be deleted. So this is showing you a substitution. Okay, substitutions usually are not that disastrous on the organism. And so you can see up here, here's our original DNA sequence. And we have TAC, GCA, TGG, AAT. So then we are going to transcribe that into messenger RNA, and this would be our sequence. And then we translate that into our amino acid sequence. Okay, but if we have a substitution, so that means we're going to change a letter out. So remember up here we had GCA. So instead of GCA, we're going to change that C to a T. And that we're going to do that at the to the DNA. So then when we transcribe our DNA into RNA, that's going to change the letter there. So instead of having G, we know that A pairs with T. And then that's also going to change our amino acid sequence. But it's only going to change this amino acid here, this histone. You can see that it was arginine, but when we change the letter from G to A, we now have histine. Insertions of letters or deletions of letters are going to have a bigger impact on the organism than just a substitution. So with an insertion, we're adding a letter in, so we're adding a whole extra letter, or with a deletion, we're taking out a letter. So what's going to happen is our sequence is going to shift, and so we call that a frame shift mutation. With a frame shift mutation, it's not going to affect just one amino acid. It's going to change every single amino acid after that letter is either inserted or is deleted out of there. So that's why we call it a frame shift. And then when we change the series of amino acids, we're also going to change the protein that we're making. So the protein could either not function like it normally would, or it may not function at all. So that could be a big problem. Okay. This diagram here is showing you an insertion. Okay, so here's our DNA sequence up here, and then we transcribe it. We get this mRNA sequence, and then we can translate that mRNA sequence, and we get our amino acid sequence. Okay, but if we insert the letter T in here, okay, we're not changing it out, we're inserting it. So we're going to shove it into here, and so then everything is going to be pushed over. So it's going to be shifted over. And that's why we call it a frame shift. So when we change, we're going to change this codon, 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 and every codon after it, which means that when we transcribe this DNA to get our amino acid sequence is going to be different. And then when we translate it, our amino acids will be different as well. So our mRNA codons are going to be different, and so will our amino acids, which means that we're not going to be making the same protein that we originally started out to make. And this is just kind of giving you an idea of a deletion is going to have the same effect. Okay, if we take out a letter, so it's, if you think of it as reading a sentence that's made up of all 3-letter words, and we take out one of those letters, like for example, if we take out that H, and then we move everything over so we still have 3-letter words, it no longer makes sense, okay? And so that's a problem, especially when you're making proteins. The second type of mutation we're going to talk about in this section is called a chromosomal mutation. With the chromosomal mutation, we're going to change either the number or we're going to change the structure of the chromosomes. So you remember we have chromosomes that kind of look like this. They're kind of looking like bunny ears here, but if we change part of this chromosome, that's going to have an effect on our organism. Or we can actually change the number of chromosomes that an organism has by, they could have an extra one, or they could be missing a whole chromosome. So we will see things in chromosomal mutations like deletions. So we'll see duplications, something that we call inversions, and then there's also translocations. So the first type we'll talk about is a deletion in a chromosomal mutation. So if this is our normal chromosome here, okay, and the letters on the chromosome, they represent genes, okay? And what happens with a deletion is we take out, we don't intentionally, but a part of that chromosome is missing. So you can see here that this B gene is missing in a deletion. So we lost all of the information that was on that particular gene is now gone. A duplication, so this time we're going to duplicate part of it. So the same original chromosome, this is a normal chromosome right here. And then what happens is, for some reason, this gene is duplicated. So there are 2 copies of this gene on the chromosome. So now we have twice the amount of information. With an inversion is a type of chromosomal mutation where we're going to switch the order of the genes in the chromosome. So again, here's our normal chromosome, and you see the order A, B, C, and then D, E, F. Okay, but here what happens is the B and the E have switched places. And we call that an inversion. The last type of chromosomal mutation we're going to talk about is called a translocation. And this is actually going to involve 2 chromosomes and not just 1 chromosome. And so what's going to happen is part of 1 chromosome is going to break off, and it's going to attach to another chromosome. Okay, so now we have 2 chromosomes. So here's our original chromosome, A, B, C, D, E, F. And what happens is you can see that part of it has changed places. So the D, E, F, what that should be here is now located on this chromosome. And this one, and the information that was located on this second chromosome is now located on the first chromosome. Okay, not all mutations are bad, okay? And there's a lot of mutations that occur and we don't even know they exist, and because they don't necessarily affect the expression of that particular gene. But there are quite a few mutations that will cause genetic disorders. So like, for example, Down syndrome is a chromosomal mutation. Babies born with Down syndrome have an extra chromosome. Cystic fibrosis is a gene mutation. It's actually a point mutation, so there's one letter has been changed. The same thing with sickle cell anemia, sickle cell disease. It's a point mutation. It's a gene mutation, and again, one letter in the genetic code was changed. And color blindness, we talked about color blindness before. Well, that is also a gene mutation. The last thing that we need to talk about in section 12.4 is a condition that's called polyploidy. Okay, polyploidy has to do with the number of chromosomes an organism has. So remember, we talked about humans they have 2 sets of chromosomes. So remember, they have a 2n number, we call them. We say that they're diploid. They have a total of 46 chromosomes, or we can say 23 pairs of chromosomes. So organisms that are polyploidy have extra sets, entire sets of chromosomes. Okay, so remember that poly, we talked about this before, poly means many. Okay, so they have many sets. So for example, our little goldfish here is what we call tetraploidy. So he has 4n numbers. So he actually has 4 sets of chromosomes. Bananas, we love bananas. They are 3n, so they are triploidy. So they actually have 3 sets of chromosomes. And one of my favorite flowers here is a pansy. And pansies are considered to be octoploidy, or they have 8 sets of chromosomes. Okay, and usually we see polyploidy more in plants than animals, but there are some plants like animals like the goldfish that do have multiple sets of chromosomes. But it's something that makes plants hardier and stronger, so it's not necessarily a bad thing.